Good Sunday afternoon, folks. Here's your next video on the cell membrane and movement across the membrane. This is the second video, and it's about osmosis, a very important process. Take a moment to write down the learning targets, please. Osmosis is the diffusion of water. So we've already talked about what diffusion is. It's the movement of a material, in this case water, from a high concentration to a low concentration, or we can say that down the concentration gradient. So it's passive because it's diffusion, and passive means it doesn't use energy. So how do we know which way water moves? Well, it depends on what kind of situation the cell is in. And there's three possible situations: isotonic, hypotonic, and hypertonic. Before we can talk about these, though, we have to figure out what a solution is. A solution is a very evenly mixed substance. Like, for example, when you dissolve Kool-Aid in water and stir, you can't really tell the Kool-Aid from the water anymore. That's how evenly mixed it is. So it consists of two parts: the solvent and the solute. The solvent is usually water. In living things, it's always water. And the solute can be common things in the cells, like sugar, like glucose, or carbon dioxide or oxygen. And there's solutions outside the cell, and there's solutions inside the cell on either side of the cell membrane. Okay? So, first type of situation, isotonic. It really helps to understand what these prefixes mean. And it turns out that iso means equal or the same. So here you got a diagram of what looks like a plant cell, and it's got that very large central vacuole here, which contains water, and you have two arrows. Don't worry so much about the color. One's going in, one's going out. So what isotonic means is that the concentration of the dissolved substance, whatever the substance is that's in question, let's just say sugar, is equal inside the cell as it is outside the cell. So if the concentration of the dissolved substance is equal, that means the concentration of water is also equal. And then that means water is going to flow equally both ways across the cell membrane. There's not any net gain in either direction. Isotonic means equal. Now, what happens if you put a cell in a hypotonic solution? This is the cell in which the concentration of dissolved stuff is outside the cell, is less than inside the cell. So hypo means less than or under. A hypodermic needle, which you get a shot with, injects some material or the medicine under the skin. Hypodermic, under the skin. So in this case, this plant cell is placed in a solution or an environment in which there's very little dissolved stuff outside. There's a lot more dissolved stuff inside. So if the concentration of the solute inside is high, that means that the concentration of the solute outside is low. Now reverse those for water. So if the concentration of the solute outside is low, then the concentration of the solvent, water, outside is high. Now I'm going to illustrate this on the board on Monday. It would be much clearer like that. So if the concentration of the dissolved substance, that's called the solute, is low on the outside and high on the inside, then water is going to flow into the cell. Now, if this is the case in a plant cell, it becomes a condition called turgid, and it just means swollen. Now, the cell won't burst because the cell wall, that big cellulose-containing structure, will prevent it from bursting. If the cell is an animal cell with no cell wall, then it's very likely that if you put a cell into a hypotonic environment that it will burst, because all it has is the cell membrane to protect it. It's kind of like overfilling a water balloon in a way. And this is called lysing, or the cell has been lysed. Finally, there's hypertonic solutions. These are just the opposite of hypotonic. Hyper means more than. So the condition outside the cell, if there's a lot more dissolved stuff outside the cell than inside, then water is going to leave the cell to try to balance that concentration. So if you were to place a freshwater plant into salt water, then that condition outside those freshwater plant cells would be a high concentration of salt. That means a low concentration of water, which means water is going to leave the cells trying to balance that up. Because remember, osmosis is the diffusion of water, and diffusion occurs because of the concentration difference. So in an animal cell, if you place it into a hypertonic environment, it will shrivel up, and this is called crenation. If you put a plant cell into that, it will plasmolyze, and basically what that means is that cell membrane, sometimes called the plasma membrane, will pull away from the cell wall. So all this yellow is the cell membrane. If you want to go back and look at the last picture, the cell membrane was pushed up against the cell wall. Here it's pulled away from the cell wall. And look at the little tiny vacuole. It doesn't have much water in it at all because it's all leaving the cell. Okay, so here are some real world situations in which you might experience a hypertonic or hypotonic or even isotonic situation. So you can read those. We'll talk about almost all of those in class on Monday. That's it. See you tomorrow.